Bring our salutations at the founder chancellor of Sri Satsai Institute of Higher Learning, Bhagavan Sri Satsai Baba. I welcome you all for this evening session. So usually we are used to having the session at 3 p.m. on Fridays, first Friday of every month. But since the speaker is from USA, so we have for this session alone, we are having the talk around 5 p.m. today. So it's my proud privilege now to introduce Dr. Ananth Vedururu Srinivasan. So I'm also happy to share that he is my PhD student as well. So currently he is working as a postdoctoral fellow in computational neuroscience at LSU Health Sciences Center, New Orleans, USA. He engaged his bachelor's and of course he earned his master's and PhD from Sri Satsai Institute of Higher Learning. Later in 2022, he moved to USA and is presently working with Dr. Carmen. He is working on theoretical and computational modeling of neurons in the medical cortex in collaboration with experimentalists from Boston University. During his PhD, of course, with me, he worked with prey-predator dynamics and optimal control problems. He was also involved in the optimal control aspects of COVID-19 within host models by the mathematical group at SSIHL. So he takes, and as well, I also take an immense pride in mentioning that he is the first graduate student from the mathematical modeling group at SSIHL, which is nothing but the Satsai Institute of Higher Learning. So, of course, that was the foundation stone was laid by that group, and he is one of, I would say, one of the person who really kind of helped us in establishing this CMB group today. So he received the academic gold medal from Sri Satsai All-Rounder gold medal. In fact, it's the Sri Satsai All-Rounder gold medal during his bachelor's. He also received his CSR Director Senior Research Fellowship during his research at SSIHL. So with this brief introduction, more maybe you can listen from Dr. Ananth during his presentation. So I would stop now. I think I'll stop the sharing the screen. So over to Dr. Ananth.
Yes, sir. Just sir, I just have a technical thing. I'll have to quit and relog in just to share screen.
Okay.
1 second.
No problem. No problem. We'll wait for you, Dr. Ananth. So just a minute. As soon as Ananth joins, we can start the session. Yes, Dr. Ananth. Can you
Yes,
share
sir.
the screen now?
Yes, sir.
Yes, it's
So
perfect.
is my screen visible, sir?
Perfect. Please go ahead.
Yes. Om Shri Sai Ram, offering my most humble and reverential salutations at the lotus feet of our beloved Bhagwan Sri Sathya Sai Baba, the founder chancellor of Sathya Sai Institute of Higher Learning. Thank you, Vamsi sir, for the wonderful introduction. Uh, good evening to all of you, and thank you so much for accommodating me at this different timing. So I will be giving you all a brief presentation today on the topic bifurcations in the brain, exploring neurons as dynamical systems. Now, I know that for a starting talk on neuroscience, there are too many details that we will have to cover. But as much as possible, I have tried to bring it in a flow where I give certain details, and uh, we can go linearly than bringing in too many other things. But I'm just giving you a small drop of the vast ocean that I'm still discovering. And also wanted to let you all know that that it's not like an expert talk as yet. I'm just 2 years into this field, and I'm also exploring alongside in this particular field of science, which has interdisciplinary aspects from almost all possible fields that I have known for. So this is the broad outline that I will just give some introduction and context, and then we will get into what are called as the single neuron models and build neurons as dynamical systems from there. And based on one of the experiments and research of my lab mate, I will give an example of this dynamical system phenomena from the hippocampal formation in our brain. And then finally, I will conclude it. So, going to give the context and introduction, when I joined or when I entered this field of neuroscience, all that I remember was from my 10th standard textbook that a neuron looks like this. It has a dendrite, it has a nucleus, it has axon, and axon terminals go and attach to other cells. And neurons specifically communicate with each other with respect to uh, electric impulses. This is all that I knew before, but when I just entered this field is when I knew that the morphology of neurons is way different compared to what we had learned before. So here are just three examples of how the cell body or nucleus that we talk about are somas. So if you look at the soma present in each of these cells, they are this dark and very black portions here, and then from there, if you look at the cell in the B, the dendrites are literally like a tree. So like that, depending on the cell body and the way their morphologies are, each one is defined for a specific need or a specific function. And just wanted to let you all know that this is a very vast field. Like we cannot say like a one shoe fits all kind of a thing. So there are so many intricacies even when we just talk about single neurons. So now, if you have to go into neuronal dynamics much before, giving you a context. So basically, as we all know, that neurons are, why are neurons different from other cells in the body? Basically, because they are excitable. So what we mean by excitable is, if you look at the inset, there is a spike. Basically, that is they fire action potential. So if they reach a certain threshold, the neurons can emit a signal, and that signal can be communicated to other neurons. And essentially, most of our body functions are due to or are a result of these action potentials that are fired. Now, if we look at some of the experimental studies previously, what they do is these are in vitro studies. That means the neurons are taken in an experimental setup after the animal or human being has died. These neuron tissues are taken on slices, and they are excited through external means. They are also called patch clampings or clamping methods, and they are given steps of current. So if you have to just look at the A figure of on the right side here, there are steps of current, and for in initial first step there is no firing at all. If you look at the second step, the firing is slightly delayed. This is the first firing, and the second, little more if the current is increased. it fires a little faster. And so basically, you see that initially this kind of firing happens depending on how much current is provided. If you look at the figure B on the same side, there is initial firing, but post that, there is also some kind of oscillation or a sinusoidal kind of which is decaying. And now, instead of giving a step current or a linear constant current, if the current is provided in the form of a ramp, and there is repetitive firing, which they call it as tonic firing. So now these neurons are behaving in different spiking patterns. Like when you give certain current, it doesn't spike at all. When you give certain current, it doesn't spike, but there are what they call as sub-threshold oscillation. That is, the whatever is not spiking. It is understood. It is you know widely known phenomena that when the current or reaches a certain threshold, or when the potential reaches a certain threshold, is when the firing action potentials are fired. So with that as a background, basically depending on different inputs, the output behavior is different. So now what could be the reason for it? Because biologically, yes, this is what is seen. So let us try to explore as to what could this be. And now this is again all single neurons being experimented. Now, I also want to give you some context of what are called place cells in our body. And this is a nice animation that was made. So what it does is, as the mouse moves in a different, whatever area it chooses, like here for now it is like a straight line ramp, it could be like a circular, or it could be, so certain neurons of our body get activated when we reach certain places. Now I will go deeper a little later. So this spiking behavior in our body for different body parts is dependent on where we are. So that way, the spiking behavior is not just about a single neuron, but that is happening day in and day out. So we need to understand what is making these neurons fire. Now, this is one part. There is another part to this. Now, we saw single neuron, we saw a couple of neurons, and this is from the mouse neocortex, and these are basically stained, and you see that all the neurons glow at the same time. So basically there is like a, we call it like all in all synchrony. Now they fire synchronously upon sensory stimulation. Like it could be that the mouse has seen something, or there is a small input that is coming. So basically, there is one, on one level, we are having a single neuron. We are right now in the neurons levels, but there can be microcircuits and networks level. And then there are lower levels where we are talking just about molecules and channels, or in the lowest level, the gene type modification, or on the highest level, behavior type modification. So now this is the span across which not just neurosciences, even the models are constructed. So I will mostly focus on just the single neurons today, and possibly if there is a little time, I might get into microcircuits and networks. But mostly we are trying to look at single neurons today. And that itself would give us an idea of how different areas of science just come together to be able to even study what this fundamental unit of the whole thinking machine in our body, if we can call it so, does. So getting deeper into the single neuron models, I would like to give you a small background of the Nernst equation and neuronal membrane. Uh, So, or if you look at on the left hand side, basically a neuron is considered as a membrane. And on the right side, we see that the membrane can be thought of as a circuit where there is inside the cell and outside the cell, the lipid bilayer, it's called, is acts as a capacitance. And there are some small pores in between through which the current or the ions pass, causing the current, and that act as resistors. And therefore, the passing of current through the neuron, through the membrane, can be thought of as a circuit. And now, if you look at the left side, we see that there are so many ions both inside and outside the cells. And so the reason for the currents or the action potentials or any form of change in the potential inside and outside is due to these neurons, due to these ions moving in and out. Now, on a leaky membrane potential, they call it. So that is, a neuron generally has small pores which are always open, but there are some pores which are open only when it reaches a certain potential. And that is when the ion movement happens on a large scale. And when that happens is when the neurons spikes. But the base of all that is basically what is called as the Nernst equilibrium equation. That is, when there is no external input or when there is no stimulus, when there is no current, what basically happens is the neurons move in and out and reach a saturation or reach an equilibrium. And for every neuron, this can happen. So basically, this is nothing but the concentration of the ion outside, concentration of the ion inside. This is the natural logarithm. RT are all the basically the ideal gas constants and the through the Faraday's law and constants. And so for each ion, we can come up with their Nernst equation, and that would give them the equilibrium potential or the reversal potential. Now, thanks to a lot of pharmacological methods, what they do is they try to block individual ions and then they obtain the Nernst equilibrium potential for each ion. And now, once we are clear that, so basically the takeaway for this is not individual details, just that the neurons is a layer and that can be formed into a circuit. But these pores that we are talking about, the ionic channels, are not just normal ionic channels. They open and close not randomly. There is a lot of science and individual neurotransmitters and way more details that get into it. And how that is done is kind of given on the left side figure. That is, we take a neuron here. Here the example is squid giant axon, and then there is a probe, and then there is another probe that goes to the extracellular reference, because we either try to keep the current constant or the potential difference constant. When you keep a current constant is when the Nernst potential or the equilibrium potential of every ion is known. So that is E, that is that potential E. We also want to know the conductance, or which is nothing but the reciprocal of resistance provided by every ion channel. So that is also measured. And then finally, there is something called as P, that is a multiplicative factor, or what is called activation or inactivation. So that is what is shown in these, just by taking the example of sodium. That so when there is a stimulation or when there is a change in potential, the ions try to move in and out. But once certain ions move in, the activation gate closes, and then it doesn't allow the ions to further enter. So the potential difference suddenly increases, and then the activation closes. Basically, inactivation kicks in, and then therefore the potential difference reduces. Now, for each ion, this changes differently, and what we see as an action potential is an overall thing put together. So here is where we need to give a lot of credit to Hodgkin and Huxley for their pioneering experimental research on this uh, animal called as the squid. And they have taken the squid's brain and then taken the two giant axons that go through its body, because it was quite huge and it was easy to study from. So they had experimented on this, and then they formed a detailed circuit. And this was when there were no technological advancements there. And in 1963, they were awarded a Nobel Prize for physiology or medicine, I believe. And basically, they come up with this four-dimensional system of differential equations. And what they have done is basically, as I showed you before, the current is the stimulus current that is provided, and they blocked each, I mean, everything else to. So if they wanted to study potassium, they blocked sodium and the other leaky channels and obtained the potential for potassium, potential for sodium, and they obtained the conductance levels and also these activation and inactivation levels. So through that, they obtained the steady state levels and then got the figures. And this is how they got the time constants and values. And so far, the Hodgkin-Huxley model has kind of stood the test of time. So a lot of recent models, they do experiments and now easily they measure these from individual neurons and then they fit them into this system of differential equations. So basically what tried to happen is the whole procedure of how the neuron is getting current in or how the currents are moving out and how the action potential is being formulated just comes through this mechanistic way of describing in the form of system of differential equations, and with these parameters which are measured from experiments, and this gives us a way of knowing more about the parameters and things directly. So this is basically like we say that now we have developed the most realistic neural network, and then you say that, oh, these are actually what I see is only equations. Yes, these are differential equations that talk about the mechanism through which the neurons behave. And now, so far, what I showed you, that equation is considering the neuron to be a point. Basically, there was no geometry. But as we saw before, the neurons have morphologies which are unimaginable, right? Which are not just small points, but they're also vast in space. So that is where advanced modeling techniques come. So the one on the left, this is called as the neurons approximation as a cable. So what we do is we so far saw just one circuit, but here we consider the neuron to be like a cylinder, the neuronal uh, the small section, and then there will be circuits which are lined up next to each other, and that is actually just again an approximation because this is a cylindrical. But if you have to actually go into the detailed morphology, you see that this is the characteristic neuron on top, and then the cable model can be, you start with constructing it more like a hemispherical side on one side to be the soma, and then multiple cylinders stacked up. So basically, this is not just individual systems of differential equations, but they are all connected with respect to another equation. And I didn't go to details for want of time. Those are the partial differential equations that connect because there is now spatial context to it. So spatially, where we measure from depends and that would give us a different potential difference or different uh, membrane potential depending on where we are measuring from. So therefore that would give us, in fact, one of my lab mates, the work that he was doing, it was 365 compartments, and luckily we have sophisticated softwares to model just one single neuron. So now getting back to this as, okay, so now we are able to describe neurons as system of differential equations. So now how do we proceed further? So here is where, again, this is the slide that I started with. That is, when you increase steps of currents, there is a different sort of a behavior coming over. And so there comes this whole thing about, okay, talking with respect to phase portraits. So now we have the system of differential equations that can tell us little more than what we just get to know from the in vitro cell recordings. So from the cell recordings, we just know that you increase current, there is behavior changing. But now through the dynamical system phenomena, we will be able to see, okay, what could be causing this change? Okay, can I use the stimulus current or the input current as a variable to see how this is becoming different, or what are the spiking properties? So predominantly you can talk about three things. So one is the resting. This is called when there are leaks happening, the ions are moving inside and outside the neuron, and at some point of time there is equilibrium. That is what is called as a resting state. Resting state is so stable that you perturb the neuron and then give it some potential, unless it has reached a certain state, it doesn't spike. So now with respect to dynamical systems, everything is becoming like a aha moment, because what would happen is someone would say, why is a neuron not firing at a certain stage? Why is a neuron firing at a certain stage? Then you would say, so basically firing is a change in the neuron's dynamical state, or there is a possibly a bifurcation happening. And if it is closer to the bifurcation, the perturbation would lead it into a limit cycle, which is giving a periodic orbit. If it is not so, it is stably sitting in its resting membrane potential, and nothing is kind of changing its values. So now the dynamical systems are giving a lot of details into what could be happening behind. So these are four predominant bifurcations that can be talked about. But in a lot of cases, even that I have you also seen, there is bistability as well. So one is saddle node bifurcation. So for just for the sake of someone who is not from the mathematical background, what bifurcation is doing is change of stability with respect to a certain parameter. That is, how does the dynamical, the phase space, or the, in this case, the membrane potential is changing with respect to, or the state of the neuron is changing when the input current or any one parameter is changing. So when the state is topologically very different, that is, in this case, basically when there is repetitive spiking, there is a periodic orbit, whereas here it's just a point. So which means the two states are completely different, and there is a parameter that is enabling this change, and that is called as a phenomena of bifurcation with respect to the bifurcation parameter that has led it to this kind of a change. So here we can see that basically a bifurcation is leading the subthreshold oscillation, which was there before. Basically, there is a kind of periodic behavior seen that is decaying, but that is getting transformed into a spiking behavior. So as I said, the saddle node bifurcation, saddle node on invariant circle bifurcation, basically another way of calling it a homoclinic orbit, a subcritical Andronov-Hopf bifurcation, and supercritical. So these are the predominant bifurcations through which the neuron goes through. There are also something called as bursting phenomena, but bursting can also come under periodic behavior and stimulus. And also, this is one another important way of classifying what kind of a neuron is with respect to the excitability. Now, there are two figures over here. One talks about subthreshold oscillations. So one is called as the resonator and one is called as the integrator. So if you see resonator are subcritical and supercritical Hopf bifurcations. What that means is there are subthreshold oscillations. That is, there is already a limit cycle that is formed, but that is not the periodic action potential based limit cycle. So if you see this figure deeply, the dotted lines is a limit cycle, but it's not spiking. Whereas the solid line is a limit cycle that's spiking. However, if you look at the bifurcations on top, the stable point is not on it is on the spiking trajectory, but basically the neuron goes from non-spiking towards spiking. So this is the original point, the one on the center, and from there it comes to the periodic orbit. So that is why it is called as the integrator. That is, it starts off in the, this is shown in class 2 excitability, basically. That is, when you give current on the x-axis, how does the asymptotic firing in the y-axis? So if you see that the frequency, the current is provided at least, let's say, until 400 picoamperes, there is no firing. But suddenly, somewhere about 450 picoamperes of current, and there is firing. Whereas if you look at the FI curve of the class 1 excitability, it is very seamless. So which would possibly say that maybe there is subthreshold oscillations already for this. So these are ways of classifying neurons to study, okay, what is the periodic firing with respect to current, and the bifurcations and this classification through that of monostable and bistable gives us ways to understand how neurons are firing. Now, why is firing of neurons important? Basically, the most of our activities like walking, talking repetitively, or most of our cognition happens because neurons spike, and they spike repeatedly. Now there is a balance in the spiking. Let's say we are going for a run. Now the running is because neurons are continuously firing and telling us to move our bodies. But then when we tell the neuron to stop, it should stop. So there is excitation, and there is also something called inhibition. So these two are like the yin and yang. Those are, go well with each other, and then they form the movements the way we would want to form. So I'll just go into a specific example from the hippocampal formation. So in our brain, there is this region called hippocampus where there are these cells called as the grid cells and place cells. Now, what are those? When initially the mice or rats had been experimented with respect to certain region. So there are two figures again here. If you look at the figure on the left, the mouse, the rat in this case, the rat was made to move in this confined place, let's say a room, and then there was one single neuron that was being recorded while the rat was moving around. And you see that there is this particular region, this region here, where that neuron spikes. So what it means is that that particular neuron in the hippocampus has identified itself with that place in the room. So what essentially is happening is the brain has actually kind of a GPS model of the place that we are located in. And these cells are grid cells. Now there are different set of cells, but what they do is, like place cells, a place cell identifies with a certain location, whereas grid cells identify with a group of locations. So these grid cell, that again the single cell, it fires when the rat was in this specific, mostly a hexagonal type of a form. So this was the actual experimental figure if you look at it. So this is a grid cell figure. So basically that particular cell fires in certain areas. So brain, there is a model in the brain that actually makes a note of this, the place. And hippocampus is known as the working memory, I would actually say hippocampus is also called as the random access memory in our body, because from there is where it goes into the long-term storage, or from there is where we can learn. So if you look at from a rodent's brain perspective, the left hand side figure is common for everyone, but this is from the rodent's figure. That is the, this is whole thing is called as the um, hippocampal formation. But what you see as MEC and LEC, they are called as lateral entorhinal cortex and medial entorhinal cortex. Here is where the grid cells are said to be, and the place cells are in this area called CA1. So this area is supposedly the first to be affected on the onset of Alzheimer's, because this is the working memory and makes us remember and takes to the long-term storage. These are the first cells that it seems are hypothesized to be affected. And also, as I was talking before, the repetitive spiking and stopping of spiking, or the inhibition with respect to the excitation, are very important. So just not wanting to get into complete details. So the red firing, the red cells here are actually the excitatory cells called the pyramidal cells, and the black or the gray traces are from the fast spiking interneurons, are called as the inhibitory neurons. So both have to work hand in hand to generate or a lot of cognitive things across the brain. And now when one of them stops, or when one is not complementing the other, it can be seen as neurological disorders, even for that matter Parkinson's. So you if anyone knows Parkinson's a little bit, basically it leads to uh, unsolicited movements in the body which were not intended to. And now there is something called deep brain stimulation and other medical cures that are coming as to trying to basically stop, basically inhibit. So this was an experiment from one of our laboratory collaborators that there was no light and there was no stimulation, and the fast spiking interneurons were spiking. And now suddenly there was a light stimulation where the instead of stimulating positively by sending current, there is a negative current, like inhibitory current provided, and it stops. Now, the experiment was to understand the dynamics and to study what could be causing this, but it has a lot of relevance in the sense that to understand, okay, what could these stopping and causing of these can be because of, because that can lead us to talk about, okay, now if there is some undesired behavior in the neuron spiking, what could be the reason? And now we have the dynamical system, which is giving us a means to go through it. So what is basically happening is, when these black traces, the gray traces, fast spiking neurons stop suddenly, the red traces seem to be more. So which means that that is not the usual thing. So basically changes the firing of the locally connected pyramidal cells. And just to give a little more information, pyramidal cells are called the principal cells. They are, some of the pyramidal cells start in the brain and come all the way down to the rest of our body, whereas these small neurons called fast spiking neurons are localized. So when those neurons, pyramidal neurons, are firing more, maybe it is implying that it is, giving some indication to the rest of the body to do something that may not be required. So to understand that, this is my lab mate, Dr. Carol Upchurch. She's now finished her medical school. She's gone back to her medical school and doing her rotations and residency now. But it was very surprising to me as a mathematician to see that an upcoming doctor was doing a high level bifurcation analysis for this model. So based on Dr. Chamberlain's work, she came up with this 5-dimensional model for this particular neuron. And if you see, the potassium currents are two. There are not just single potassium current. There is sodium current, potassium current, sleek, and stimulus currents. And how do we fit the model? So now you have come up with a dynamical system or differential equations based on experiments I was showing you, right? These are the frequency injected current curves. So as the current increases, the frequency changes, and those are captured. Now the red trace is basically for this particular neuron, a single neuron that fired in a certain way. And how do we validate the way model works is see how model and experiment work together. So the purple trace overlapping with the red trace is the model that we are looking at. So the model, the red trace being the model, and the purple trace overlapping with the experiment. So for that portion is where our model is actually, realistically replicating the experiment. And then once we know that, we now use the model to study a lot of other things. So what she did was, now the experimentally also, he was trying to see what could be the reason for this interruption. So here there is an input and suddenly there is an interruption of firing, and it gets back to firing back in the same mode. So now he wanted to experimentally check that what could be the reason. So he wanted to see if there are, as I told you, there are two potassium currents. So he wanted to see if KV1 could be a reason for it. So again, thanks to pharmacological and a lot of developments, that once he blocked the potassium blockers, then for the same disturbance, the firing did not stop. That is shown by the pink traces. So in the black traces, you see that the persistent firing is interrupted, whereas in the pink traces, it's not interrupted. So there is some indication that the persistent interruption could be due to KV1. But to mathematically prove it is what my lab intended to do. And then this was done by looking at this H flow, or the uh, slow variable due to the potassium. So basically, this is an activation inactivation parameter that is showing how basically the inactivation. This is the change in current, and it's inactivating very slowly. So how was that possibly the reason for the persistent interruption? So what it's being shown is, there are two solid curves, that is the voltage minimum and voltage maximum. The y-axis is voltage and x-axis is the bifurcation parameter, and then the stable and unstable values. And there is, and basically it's showing that there is bistability happening. Now, in this case, that is, there is a spiking limit cycle and there is an unstable limit cycle. The unstable limit cycle is leading towards, is coming from the Hopf bifurcation, and the stable limit cycle is a persistent firing. So when the input is provided, it is somehow coming back from stable firing to unstable firing, and it goes into quiescent state. So that is what is being shown through the bifurcation diagrams. Although this was not theoretically proven, but experimentally and numerically, it was shown that these bifurcations are what could be causing the change in firing patterns. And this was very important finding, because this kind of analysis now cannot be done on an experimental setup. So this is where, in this field, the back and forth thing happens. So when a modeler, when an experimentalist does some analysis and then gets back to a computational person will be able to do with respect to every variable, not just in the injection current, because the dynamical system is at his or her disposal. So basically, this was shown that there is bistability occurring in this phenomena. And so again, now to say, now we can, if you have to look at the larger picture, basically we can say that the firing was stopped because of a change in bifurcation, because the dynamical state of the neuron was drastically changed due to an input. So essentially, some of our cognitive states are due to different bifurcations of neurons in the brain. And to me, when I came from a mathematical biology background, was one of the most exciting things in this field. So I'm not too sure how much more time, so I will just provide interim a conclusion towards this, and maybe if there are any questions, I can go deeper into some other things. But uh, this is one of the quotations from Eugene Izikovich, and he says, information processing depends not only on the electrophysiological properties of neurons, but also on their dynamical properties. And even if two neurons in the same region of the nervous system have similar electrophysiological features, they may respond to the same synaptic input in very different manner, and that is probably due to the each cell's bifurcation dynamics. So the bifurcation plays a different role, a very important role in being able to see how the neuron responds to a given input and what is the result of this. Now, I just wanted to give kind of end on a very general statement. It's overall about, in a research that was done to study the neocortex, or so what is neocortex? It is basically that the humans have evolved to, which are not in animals before. It is very less in the animals across evolution, but for humans, the cortex is the uh, epitome of intelligence, quote unquote. So when they are pursuing to study cortex, the cortex is a model building system. It essentially learns a model of the world and the model of everything in it. So what the researchers came up was that basically the neocortex, or the intelligent part of the brain, which is not, basically they say the small brain or the lower brain and the higher brain. So the lower brain means that which we use, which us as humans and animals have completely in common, whereas the neocortex is something that is new. New means new. So that is something that has come for humans to be able to apply intelligence across fields. And now when we say artificial general intelligence, we are trying to see how we are how we can bring in that intelligence into computers as well, because as of now the intelligence we talk about AI are just through the input output black box neurons, which are inspired by biological neurons, but a lot of the things that the biological neurons do are actually nowhere done by the AI models. So therefore, this is a research by the company Numenta. He's also the founder of Redwood Theoretical Neuroscience Center in California. So they are doing complete theoretical research based on experimental results on how is actually, how can we reduce the artificiality in the artificial intelligence. And so the theory that they have come up with is that the neocortex in the brain are actually the samples of the same thing that is repeatedly connected together. So what we call visual cortex, motor cortex, sensory cortex get same inputs in the form of spikes. So when I'm talking, or when I'm seeing something, when I'm hearing, or when my response to touch is all spikes. So basically, the brain is getting all the inputs in the same way, and it is giving outputs also in the same way. It's just that the different body parts are doing different things. So, which is very similar to the CPU phenomena of the computer, right? That is, you have a single processor that processes it in the same way and gets back. So therefore, the theory that they are coming up with is that every small unit in the brain is actually doing the same thing. And in fact, they go to say that basically what we are doing in the brain is actually modeling the world. So what I'm seeing is the model that my brain has made to see, because actually we cannot see all color, I mean all frequencies, we cannot hear all frequencies. So everything is basically a model, and every time our brain has a voting system to say which model is right. That is basically when we see optical illusions, we see one thing, but when somebody corrects it for us, we are able to correct it and then get back to what we think we saw. So that is basically the brain doing course correction at every instant, saying, okay, what is right and what is wrong. So essentially what it's trying to say is that the incorporation of space, time, everything is all in your head. When they say it's all in your head, he says it's literally all in your head. And here is where another quotation, and from who, all that you find in this world is a reflection, reaction, and resound. Do not get carried away by them. Aspire to attain reality. In order to experience the reality, give up body attachment and try to understand the divinity within. So, because when I read this book by Thousand Brains Theory by Jeff Hawkins, and when he said everything that we are projecting outside is actually what is from within, I completely connected with what founder chancellor Bhagwan has told us about reflection, reaction, and resound. That everything that I am hearing to is, if I'm hearing to someone speak, it is my simulation of their voice. So, because sometimes when we hear, we say, oh, they sound similar. That is because your brain finds it similar. So, with that thought, brain being one of the most complicated, organs, although there has been so much of pioneering research in the field, there is still lot more to understand about it. And currently, what I'm working on are network properties of neurons in the terms of pulse coupled oscillators, and we're trying to understand how the theta gamma oscillations and fast wave ripples, are actually founded in the hippocampal formation or the medial entorhinal cortex. And this acknowledgements are to LSU Health New Orleans and my PI, Dr. Carmen Canavier. And this is also a NIH sponsored project along with John White, who's our experimental collaborator from Boston University. And uh, I thank the whole lab for taking me in as complete novice to the neuroscience field and helping me train and come up to wherever I'm able to come so far. And immense gratitude to Satyasai Institute of Higher Learning, DMAX, which is the home from which I've come so far, and Bhagwan for all the blessings and for his constant guidance and support. And I thank Dr. Vamsi and CEMB for giving me this opportunity to discuss my research with you all today. And I mean, overview of some research that we do. And thank you all for your attention. Any questions? And you can contact me on my email.